the next part of the lecture will be. So now the next part lecture is quite different, which I am going to include today. So I am going to present one of my own review paper, which is quite good uh, audiences and readers, and also, of course, citations in less time. It is becoming quite common to go for pharmacogenomics field. So this was a review paper which I published as a corresponding author. Here, in the current challenges, especially in the complex diseases like human cancers, diabetes, and some more human diseases, the people are focusing on understanding the genomics for the patients and then look for the solution in terms of pharmacological approach. So the pharmacogenomics means genomics plus pharmacology. That is what it makes it pharmacogenomics. So the title of my paper was A Genetic Approach to Drug Development and Therapy. So it will cover also pharmacogenomics overview and goals and how to understand the personalized medicine. The other purpose also, it will help you in another way if you want to write a review paper. So you need to, how you need to proceed. For this purpose, you need to choose a, an interesting topic which you are familiar with and then try to find few major points. For that, you need to read the leading and high impact research and review papers with recent publications, and then make few points, major points, which you would like to address. And that is what makes your paper as good and with less rejection chances in any international peer-reviewed journals. Now, what I am going to introduce, so the abstract was quite normal, not too short, not too big. But as I mentioned, that this pharmacogenomics review paper was meant for understanding the research used in medical sciences which contributes to the understanding of medication interactions. And the purpose of this work was to provide a review that might serve as a foundation for the creation of new pharmacogenomics application research publications, techniques, and strategies. So the main keywords or field I have covered is pharmacogenomics, genetic approach, drug therapy, drug development, and personalized medicine in human diseases. In introduction, you need to get the main top area to introduce them in sequential way and then move for different sections, different goals, what you are going to address and how you are going to address and what are the main topics which you will be subtopics which you will be including for this review work. So here it was quite simple to understand my review paper that I was interested in clinical sample collection, genotyping, associated loci, epigenetic effect of variants. And then, so basically it is saying that the summary of steps taken to pharmacogenomics therapeutics. It starts from integration of multi-omics data and proceeds through pathway level understanding, pathway level interaction, and network level understanding, unraveling the integrated mechanisms and predicting the optimal putative biomarkers in case of human cancer. So if you understand this layout, this will be quite easy for you, even if you are interested to work on the research topic or review topic, or if you are working in any non-academic job, or let's say private organizations, for example, pharmacological company or pharma companies. So you need to understand the implementation of precision medicine by including the next generation sequencing methodology and technologies, and of course, the computational softwares or tools developed for this purpose. So, the computational techniques or data from previous research can also be utilized to train and predict the functional consequences for the disease, selected disease, if it is not fully understood. So, what is the main purpose here? If you want to understand how pharmacogenomics works, then you need to think, collect the tumor samples if we are talking about cancer, or collect these samples, then look for the genomic compositions and the aberrations with respect to the normal, and then look for different kinds of treatments in the presence of the drugs, different drugs, and then go for the search of the optimal drugs for the that specific individual or patients. And of course, you can look for the variation in the treatment outcomes, pharmacokinetics, germline genome, and then finally you will have some kind of therapeutic drug. So pharmacogenomics is a field concerned with the impact of genetic changes that determine an individual response to a specific medications or drugs. And what medications pharmacogenomics help to evaluate? For example, here you can see blood thinners, body stabilizer, chemotherapy, anesthesia, drug antibiotics, pain medications, and cannabis. So here you need to understand the factors associated with the problem, or that is drug response. So how the drug response will be by after the treatment of a specific drug in case of cancer. Look for genetic background, genetic heterogeneity, mutational patterns, epigenetic regulations, and environmental factors. These are the main factors and which you need to understand. Then what are the future of genotypes in drug therapeutics? So here you can think in multi directions. If you so future mainly of genotypes in drug therapeutics is precision medicine. And how precision medicine works is multiomics analysis, specific biomarkers prediction, then personalized drugs, a specific dose, personalized diagnosis is done. This is summary of it. But if you want to understand with real application, then here you have cancer immunotherapy. You can look for this, and this is from one of the review paper, which you can see in Nature Review Drug Discovery. This is one of the best example for going for the personalized therapeutics in case of cancer. Here you have the details how you will be looking for the personalized therapeutics by using immunotherapeutic approach. Now it comes drug response, how you look. So basically, the response after the drug is inserted or the patient is treated with the drug, how the patient basically, you can say, response. If there is good improvement with less side effects, or good improvement but high side effects, less improvement but high side effects, something, many more combinations. And that is called the drug response, which you need. And normally, the researchers look for that drug response in the form of the curve uh, plots. So this is called dose response curve also. But if you are a patient and you are not familiar about these dose response, you just need to ask yourself if you are improving more or deteriorating more. This is what you need to think as a layman. Then you need to understand the genetic factors associated with phenotypes. As I mentioned here, for example, genetic factors associated with phenotypic variabilities, these are five things mainly: genetic background, genetic heterogeneity, mutational patterns, epigenetic regulations, environmental factors. But here you have the details about those things, for example, sequence variations. Then you look for SNPs, insertions, deletions, or this that is also called indels. Then structural variations, that is also here again indels. So in structural variation is also called as SV. That means here there will be the deletion or insertion on chromosome, which will lead to the complete change in the chromosomal structures. Then inversion, duplication, and translocations are also there. So then from type to phenotype. This is what you can understand. Gene environment interactions, epigene environment interactions, gene environment interactions, how you can think about genetics and environmental factors, then what are the epigenetic changes, and finally, what is the disease or phenotype. Then look for the genetics and the environmental factors. Then again, look for the epigenetic changes and phenotype. So there are multi aspects in which you can look for it. Then you need to think what are the SNPs associated, what are the methylation and histone modifications associated, what is the, if you are looking for the environmental factors, then nutrition, stress, toxins, and pathogens, and the exposure of the patient to certain environment, you can also look for it. Now it comes to the knowledge gap about the genetic contribution to phenotype variation, or you can also say the barriers in achieving the final goal for the personalized medications. There are a number of challenges and knowledge gaps still persists. So you need to think about the physiology, ecology, phenology, biochemical networks, cellular functions, or you can say the deep knowledge of system biology, of real biology, instead of just computational biology, protein function and interactions, identification of the selected genes, environmental associated analysis, and then you think about the environmental things, fitness and phenotype, and you can play with the computational coding and factors and the algorithms used. So you can look for the inter and intra specific competition, adjustment of trophic interactions with grazers, resource allocations, maintaining genetic variation and diversity, then genetic response, and finally, you can look for the implications. Now, in my paper, I have presented only the figure number 3, which you can see about the epigenetic effects and regulation of gene expression at the mRNA and protein level, but you can look for multi-direction in multiple way. So this one was basically, I tried to mimic from the published review paper, so that I have redrawn this thing in PPT, because in review paper or in your publication, you cannot adapt the as it is figure from any published data or the others data. So you can redraw it instead of using them as it is. Now you can look for example, if you are thinking about this aspect, you can look for the mechanistic models, you can look for the variants, master transcription factors, and then coding and non-coding reasons, role, promoters and enhancers, how they could play the potential roles. So I have summarized, if someone is interested to develop the computational approach for the research purpose for precision medicine or pharmacogenomics, then this could be the step where I have summarized them in 5 steps: data collection, multiomics data analysis and integration, systems biology approach, experimental validation and evaluation, then final assessment and conclusion. So this is the summarized way for looking and understanding about pharmacogenomics and precision medicine. If someone is interested, what are the phases in which it could work? So there are 3 main phases: phase 1, 2, and 3. So inform, discovery, candidate genes, drug metabolism. Then phase 2 includes primary discovery, genome void associations, whole genome exome sequencing, and then you can look for the phase three, that is the trials, stratified trials, continued discovery, and what are these discovery, genetics, oncology. These are the different things you can think. So predictive things for the response, resistance, new drug combinations, multiple drug combinatorial therapeutics, and new drug target predictions. These are the tables which shows you the basic molecular biology methods required for doing these things. And if you are just going for computational part, then you need to look for these basic softwares and databases from where you can get the detailed information required for working in pharmacogenomics field and developing your own models. Now, finally, you can also think about basic sequence analysis as it was being carried out 10 years, 15 years ago, and predictions and the functional impacts of variants. You can think about them now. It is a little bit revolutionized because of the advancement in high throughput sequencing, that is NGS. So you can now think about the motif signatures, gain or loss of functions in different way. While earlier, 10, 15 years ago, the people were mostly focusing on sequence alignment, is that. Here, of course, you do all those things, but now it is quite basic things. And then develop certain kind of model, train them in order to predict the phenotypes very close to reality, and then look for the regulatory reasons or variants about promoters, enhancers roles, aberrant splicing, 5 prime, 3 prime UTR's role. What could be these roles playing these five things: cis regulatory element, promoters, TAD boundaries, coding sequence, and untranslated reasons. And finally, you can move towards thinking about the real perspective of drug. So you need to think also about the structural data. You need to think about the functional effect, genetic variations, drug and dPCR targets, then cells information, and then look for the dose response curves, of course. Here, what are the benefits of this kind of pharmacogenomics approach? It will help you in early diagnosis, more precise diagnosis, more effective treatments, fewer side or adverse effect or reactions, eligibility for clinical trials, prognostics, and preventive approaches. Now, you can summarize them. So, why we are preferring pharmacogenomics or personalized medicine approach in with in instead of the domestic or traditional approach. So here you can see it was more time consuming and more cost things. But now you can carry out the new drug development or discovery in just few months to few years only. So these there are huge benefits of precision medicines which you can use about it. Look for it, and then finally summarize the approach which you can think about the new paradigm, high success rate, low cost, more effectiveness, drug development driven by human biology. What are these things? What is the how should we look for the genome, lifestyle, nutrition, epigenome, microbiome, age, gender, BMI, relationship, drugs, and many more things.